It's my great pleasure and honor to welcome you to the Translational Workshop on Cancer Immunology Discovery Approaches into Clinical Trials. My name is Desmedt. I'm an Associate Professor and Head of the Lab for Translational Breast Cancer Research at the KU Leuven in Belgium. I will be the moderator of this workshop. Today, we have an excellent panel of accomplished investigators who will share examples of technological advances that have driven important clinically relevant discoveries in the field of the tumor immune microenvironment, and more especially in the field of triple-negative breast cancer. We will first have all the presentations, so keep your questions also for the end, or you can always put them in the chat. So the first speaker for today will be Dr. Raza Ali. Dr. Ali is leader of the Systems Cancer Pathology Group based at CRUK Cambridge Institute at the University of Cambridge. He's also Associate Director for Clinical Academic Training and also a honorary consultant pathologist at the Addenbrooke's Hospital in Cambridge. He read medicine in Cardiff and began his training as a pathologist there before moving to Cambridge to undertake his PhD in the quantitative pathology field and genomics of breast cancer under the supervision of Professor Carlos Caldas. He completed his specialty clinical training in Cambridge before moving to the University of Zurich. And in Zurich, he worked under the supervision of Professor Bernd Boden-Miller, who is the inventor of imaging mass cytometry. And there he conducted research using highly multiplexed epitope-based tissue imaging to understand the principles of spatial organization that characterize breast tumor ecosystems. So please welcome Dr. Raza Ali for this first presentation. Thank you.
Thank you, Christine, for a very kind introduction, the main message of which is I need to make it much shorter. So delighted to have you all here. I'm hoping to start the presentation shortly, although I'm guessing that you can't see it because I can't either. I'm going to be talking about multiplexed imaging for precision breast oncology. These are my disclosures. And in particular, I'm interested in breast cancer and immunotherapy because it really places some challenges on us as treating clinicians. How do we really use immunotherapy most effectively? I'll talk a bit about the technology, the principles of how it works and why it works the way it works, because I think it's important to understand that, before talking about the main application, which was to use this technology to discover single-cell spatial biomarkers of immunotherapy response. And then I'll talk a bit about the potential and particularly the challenges, many of which will become obvious as I speak. So this is a summary of the results from the KEYNOTE-522 trial, which this crowd will know extremely well, which is a great result for patients with early triple-negative breast cancer. There's a material extension in survival with the addition of pembrolizumab. In the publication, there is also the subgroup analysis, which I think is particularly informative and worth paying some attention to. And it's worth paying attention to for what it doesn't show us. It doesn't show us any subgroup which derives differential significant benefit, and that includes patients defined by the canonical biomarker PD-L1. In other words, patients with PD-L1 negative tumors derive comparable benefit from the addition of pembrolizumab from those who had PD-L1 positive tumors. And it's similarly clear that not all patients are benefiting equally. So we have a challenge on our hands to how best to use this expensive and, of course, possibly very toxic medication. So what could we do? Well, we could go and look at the immune checkpoint blockade biomarker field as it currently stands. It's a large field of research, particularly in non-small cell lung cancer and in melanoma, and see what they've done and ask whether this might be useful in breast cancer. This is the result of a meta-analysis published about almost 5 years ago, where they did something very simple, which was to compare a handful of the most popular biomarkers of response to immune checkpoint blockade across tumors and to ask how they performed and what their relative performance was. So what you see is that PD-L1 immunohistochemistry and gene expression signatures, typically of T-cell infiltration and tumor mutational burden, all performed comparably, although they're probably complementary. But what you see there is a light blue line, which was based on a relatively modest amount of data for multiplex immunohistochemistry and multiplex immunofluorescence. And this is really an indicator of the possibility that these sorts of approaches may outperform technologies based on a summary statistic or on bulk tissue analysis. And so this was of particular interest to me as a pathologist, and particularly as somebody who's interested in characterizing the TME in intact tissues. So how might we do that? What kind of multiplex technology might we use? The one I'm going to be talking about is imaging mass cytometry. I'll describe this in some detail. I couldn't go through all of the technologies because there's been such an explosion of them, but I'm sure many of you are familiar with other approaches like cyclic immunofluorescence. Imaging mass cytometry is essentially an antibody-based, principally an antibody-based approach, which allows you to do many things, to target proteins as well as post-translational modifications. But essentially, since what you're interested in are rare earth metal isotopes, that's the reporter rather than the fluorophore, anything you can label with a rare earth metal isotope could be used as a method to probe the tissue. So we've used both antibodies and branched DNA to target transcripts in situ simultaneously. And typically nowadays, we're looking at about 44 channels simultaneously, so that's more or less the upper limit. That's really limited by the number of pure isotopes that are available on the market. The technology would allow you to go further, but as I say, the limitation is how many reagents are available. And essentially what happens is you label your tissue simultaneously with a cocktail of antibodies. You then ablate that tissue using a 1-micron laser. Each resulting tissue aerosol is extracted, is pushed through inductively coupled plasma to ionize the isotopes, and their abundance is then quantified using time-of-flight mass spec. And all the data, that means, are generated simultaneously. You're not doing any imaging cycles. Having said that, this is far from a perfect technology because you need a dedicated instrument, which is almost $1 million. It's quite time-consuming. It's not a rapid technology. Whole slide imaging is at the moment difficult to do, at least at 1-micron resolution, and it is on average expensive. Having said that, we do derive very reliable and high-quality spatially resolved data across these different epitopes. And these are pixel data, which are computationally re-registered to their original position, and then we subject those to a series of image processing steps using a combination of data-driven methods and deep learning. That includes segmenting single cells, classifying those cells as being either epithelial or in the TME, and then classifying those further into different subsets. And that allows us to ask questions about the cellular composition of a sample, cell-to-cell interactions based on physical proximity, as well as higher-order interactions in the form of multicellular structures. So that's the technology. What did we do with it? We asked this question. We asked, what is the relationship between TME structure and immunotherapy response in triple-negative breast cancer, in collaboration with Luca and Giampaolo in the neoadjuvant trial in triple-negative tumors. Giampaolo and Luca had collected samples at 3 time points, remarkably, for almost a very large proportion of patients. These are formalin-fixed paraffin-embedded samples at baseline, so those are treatment-naive samples, after 1 cycle of treatment and after treatment. And we subjected these to IMC using a panel of antibodies designed in particular to understand T-cell activation states, although not exclusively, and use pathological complete response as a surrogate biomarker of the endpoint for this trial. Some of you will notice that carboplatin is on this list. There's no antibody to carboplatin, but I'd like to talk about it because it illustrates a principle of how this technology works and also a small fringe benefit. And what we did for carboplatin was just to leave one of the platinum isotope channels open. Carboplatin is, of course, a platinum salt, and so the theory was we should be able to detect endogenous carboplatin directly in the tissue. And that's what's summarized in this box plot. So in gray are patients who received just chemotherapy. The orange ones are those who received immunotherapy as well. The x-axis shows the 3 time points. B is baseline, OT is on treatment, PT is post-treatment, and the y-axis is the amount of carboplatin that was detected. So if you look at baseline, there is some distribution. That's essentially the noise in the data. But what's interesting is that after 1 cycle, there is clearly an increase, but after the full treatment course of 8 cycles, that increases further. So there's a dose-response effect that which we can detect, and which was unexpected and rather gratifying. Of course, these are imaging data, so we should be able to see where that carboplatin is accumulating directly if the image actually appears. Oh, there we go. Yeah, so we can in fact localize the carboplatin, and principally it seemed to accumulate in macrophages. You can see there with co-localization with CD68. So this is a nice benefit of the technology. As I say, it really explains how the technology works by quantifying isotopes directly in tissues. And in this case, that isotope was a drug. But we wanted to make more sense of this, of course, and so we subjected these tissues to the sort of workflow I mentioned earlier in the presentation, and classified cells into epithelial, i.e. cancer cells, and TME cells into 16 and 20 phenotypes respectively. We did this very carefully. We mapped these phenotypes back to the morphology in the tissues, which is an extremely important point in understanding the fidelity of your data and ensuring you're not making serious mistakes in cell classification. One of the things we were really interested in was cell-to-cell interactions, because we're trying to identify biomarkers of immune checkpoint blockade, which disrupts a receptor-ligand interaction, which requires cells to be physically in proximity, as well as, for example, cytotoxic T cells needing to contact their target cell through the T cell receptor, which suggested to us that cell-cell interactions might be important. We classified these very simply as whether two cells were physically touching. In other words, they had to have at least one contiguous pixel in order to be defined as interacting. And we did this in a specific way. There's no obvious intuitive way of doing this, I should say. It sounds like it's straightforward and there should be a very simple way of doing it, but you really have to think and make decisions about what you want to do. And so the way we did it was to define two types of interaction: homotypic interactions, illustrated here, between cancer cells, and perhaps more interestingly, heterotypic interactions between cancer cells and TME cells. And so we computed for every TME cell phenotype the number of interactions. So that's the number of cancer cells each TME cell is touching. So one cell can, of course, touch more than one cancer cell, and normalize that for the total number of cells segmented, and then ask whether that was predictive of immune checkpoint blockade response based on treatment-naive pre-treatment samples. And those results are summarized in this plot, which are the results of logistic regression analyses. So again, orange are patients who received immunotherapy, gray is those who just in the chemotherapy-only arm. And what you see is that we were able to detect several markers of response, particularly in the TME interacting with cancer cells, which were markers of response of patients who received immunotherapy. I'll highlight just two there, which show the largest effect. So one of them is B cells, so CD20 positive B cells, and their physical interaction with cancer cells, which is in itself somewhat curious, because that doesn't really make a huge amount of biological sense. I think what we're detecting here, and I think this is an important cautionary note, is an association based on a state of organization of the immunotherapy, of the immune response rather, which is indicative of sensitivity rather than a functional interaction. I mention that because that's contrasted by activated T cells, so CD8 T cells positive for granzyme B, where a physical contact with a cancer cell is more likely to be functional. And so we investigated that further using a very simple analysis. We defined T cells as being in touch with cancer cells or not in touch with cancer cells, and asked whether those touching cancer cells showed a differential activation state. And what we saw was for both cytotoxic T cells and T helper cells, that T cells touching cancer cells tended on average to have elevated levels of activation markers, be it TOX or PD-1. But moreover, they also tended to be much more proliferative. And in fact, this was a surprise finding because the extent of that effect was really quite marked, suggestive of clonal expansion. That also highlighted the utility of a simple marker, namely Ki67, to identify with high fidelity activation states of cells in tissues. And so that redirected our attention to these original cell phenotypes. Many of you will notice there's a gray bar here next to the different cell phenotypes, which is indicative of the proportion of that cell phenotype which is proliferating, the proliferative fraction. I highlight it because it's markedly different both among cancer cells and in the TME. So as you might expect, the proportion among cancer cells is higher, but nonetheless, there are certain cell phenotypes among just triple-negative breast tumors, it should be recalled, that show proliferative fractions of less than 5% versus others which are in excess of 50%. Likewise, in the TME, there are important gradations, some cell types in less than 5% and others in excess of 15%, particularly some T cells, but also some antigen-presenting cells. And so again, we asked a very simple question: whether these proliferative fractions computed as the proportion of a given cell phenotype positive for Ki67 were likely to be predictive of immune checkpoint blockade. And we did that both for cancer cells and for TME cells. And now we started to see some things which hadn't been apparent in the data when we didn't look at it this way. First of all, we found markers of immune checkpoint blockade among the cancer cell autonomous compartment. So cancer cells which showed aberrant expression of MHC2, as well as at PD-L1, their proliferative fractions were significantly associated with response to immune checkpoint blockade. The TME really showed quite a dramatic effect. I think there are a couple of messages I'd like to emphasize here. First of all, that a proliferative TME in general seems to be indicative of sensitivity to immune checkpoint blockade. Most cell phenotypes were associated with response, with the exception of some which you might expect, like endothelial cells and, for example, neutrophils, which didn't show a significant response. But nonetheless, there's a gradation, and CD8 TCF1 T cells showed the greatest and strongest association. And I think that makes some sense because these cells are thought to be a progenitor T cell type, which provide the proliferative burst following immune checkpoint blockade, and that's been shown in other settings. Again, this was interesting, it was notable, and we wondered whether these were just the same tumors. In other words, were tumors with high MHC2 proliferating cells also the same tumors with high CD8 TCF1 proliferating cells? And there is some extent to which those two things are correlated, but that correlation is modest. So, in fact, the picture is rather more complicated, and these things are complementary. As you probably noted, what we've got here is a collection of different biomarkers based on abstraction at different levels of these tissues, and there are many of them. And we really had a question, which I'm sure is in the minds of many of you now, which is which are the most important, and how do we interpret them? And so the way we chose to do that was to use a method well established in machine learning for feature selection, which essentially involves permuting your data and asking to what extent does that permuted variable compromise the ability of a model to predict outcome in order to rank these different features. We started with about 150 features and ended up with about 10% of them, so about 14, which survived that analysis, so only a small proportion. Moreover, of those, these 4 were doing most of the work, and these are some of the usual suspects. So these include the proliferative fractions I mentioned. They also include some of the spatial interactions I mentioned. The other thing I think that's worth noting here is that these biomarkers are both in the cancer cell autonomous compartment and in the TME. They're indicative of cell activation states and of spatial architecture. So all of these different facets of the tumor are likely to be important in predicting response, and it was nice to see that they were complementary. Having said that, if you look at the heat map on the right, what you'll notice is that the bars underneath the word PCR, in other words, responders, are all brighter than the bars on the left, which are labeled residual disease. And so what we discovered was the same sort of thing that everybody discovers more or less in immune checkpoint blockade, which is markers of response. In other words, positive predictive markers. The things that we found were all higher in patients who responded rather than in patients who did not respond. And we were really eager to see if we could discover a marker of resistance. Unfortunately, at the baseline sample and the pretreatment samples, we didn't find any. There were no markers which were elevated in patients who are non-responders versus responders. But in the on-treatment arm, so this is, I remind you, after 1 cycle of therapy, we found that cancer cells which had elevated expression of CD15 really stood out as being enriched in resistant tumors. So these were associated with residual disease, eventual residual disease. And this was, in fact, the only marker of ICB resistance in the study. Why are cancer cells expressing CD15 associated with immune checkpoint blockade resistance? I don't know, but I will speculate. First of all, if we look at their, if the image comes up, look at their distribution in terms of their expression profile, what we saw quite often, not exclusively, but quite often, was this discrete pattern of expression. So it really looked as though cancer cells had opted to take on a different state. And it looked like this was a discrete state relative to adjacent cancer cells. So these clearly are CD15 positive, they clearly are cancer cells, but they are contrasted by their adjacent cells, which are completely lacking CD15 expression. So what could it mean? Actually, at the time the paper was published, I didn't know. I still don't know, but I'll speculate based on some more recent findings. So recently we've been looking at normal breast tissue, and what you see here are some H&E images of secretory cell change in normal tissues. These are not women who are lactating, but just have background secretory cell change. And what we find is that those cells, particularly luminal secretions, as well as the apical surface of those cells, and sometimes also their cytoplasm, are highly enriched for CD15. So to me, this suggests that this is a metabolic state. It may be a metabolic state which is taken on during lactation, and it's conceivable that it's associated with immunosuppression. But that is, of course, speculative. Hopefully this will move to the next slide at some point. Oh yeah, so the paper is published, and there's lots more in it, so do feel free to have a look. The data are also free to use for academic use, so I'd encourage you to do that. Yeah, so we're doing some additional follow-up studies to understand some of the points I mentioned. So CD20 B cell interactions with cancer cells, we're trying to understand their association with the spatial landscape of B cells. And so we've designed a panel of antibodies looking at looking at both proteins and RNA simultaneously, and managed using those data to classify 6 subtypes of B cells, and we're currently characterizing their associations with response. Likewise, to understand these platinum-laden macrophages, we've designed this panel to look at the myeloid compartment to really define it much more cleanly and with greater fidelity and look at their activation states. And we're also finding these large differences in the amount of platinum accumulated in those cells and different subtypes of cells, and also that differs by response in black versus lack of response in gray. So this is ongoing work. There's lots more to do. So briefly, tumors are complex, but ultimately they are structured, and I think that structure can be parsed by appropriate analyses. These sorts of clinical studies, particularly using technologies which allow enumeration of different cell types, I think a great complement, particularly for preclinical studies. And because multiplex tissue imaging is based on epitopes, based on antibodies, in other words, it's quite an effective way for pragmatic discovery of biomarkers which could be used in a clinical setting. Having said that, we need to figure out ways to rank those biomarkers because these technologies result in lots and lots of data. I've shown you one method for doing that. The other issue is, how would you ever get this into a clinical pathology lab? That's a major challenge, particularly if you're going to use multiple markers, and also depending on how you want that to be approved. Of course, we've used commercial antibodies, and that's one challenge itself, as well as there being several others. So just to thank my collaborators, Luca and Gianpaolo, my funders, Cici and Lubna, there highlighted, did most of this work, and thank you all very much for your attention.
Thank you very much, Raza, for this beautiful presentation. So the next speaker of this session will be Diether Lambrechts. He's a professor at the KU Leuven in Belgium, and he's also the head of the Laboratory for Translational Genetics. It is a technology-driven translational research group. And with his team, he aspires to tackle actually important questions in oncology by translating genome-scale datasets into clinically applicable knowledge. He's also a group leader in the VIB KU Leuven Center for Cancer Biology and also a professor at the University of Leuven. Dr. Lambrechts was also trained as an engineer at the University of Leuven, and he initially also worked under Peter Karmelit on the role of vascular endothelial growth factor for his PhD. He then also worked at the Wellcome Trust Center for Human Genetics in the University of Oxford, England, before joining VIB as an independent VIB group leader. So today we are very happy to welcome Dr. Lambrechts for his view on this of triple-negative breast cancer and the importance of the tumor microenvironment. Thank you.
So thank you, Christine, for the nice introduction. So I will be talking about how our lab is using single-cell technologies in order to study, to do our breast cancer research. And so when we compare against bulk RNA technologies, single-cell RNA sequencing technologies really stand out because they allow you to study the heterogeneity that is within the tumor microenvironment. Here on the right side, you see, for instance, how single-cell RNA-seq can identify up to 17 different T-cell subsets in the tumor microenvironment using single-cell RNA-seq. And if we look at immune cells in general, this heterogeneity is even larger, with up to 72 different immune sets that can be identified using single-cell RNA-seq. We are also using a lot of single-cell TCR-seq, and so this technology really allows you to trace immune-reactive T cells within your tumor. So here you see on the left side how single-cell RNA-seq can identify heterogeneity in the CD8 positive T cells. So these CD8 positive T cells can differentiate according to 3 different trajectories. It all starts with naive CD8 T cells, which can then differentiate, for instance here, towards the CD8 positive exhausted T cells. And while they do so, you see that their expression is slowly increasing. So they upregulate interferon gamma as a sign of T cell activation, but also cytotoxic markers are increasing. And then finally also these typical exhaustion markers are increased. And at the end of such a trajectory, when you have these exhausted T cells, it basically means that these T cells are expressing PD-1, but also still expressing these cytotoxic markers. Now, when we look at the TCR sequencing data, we see that these naive T cells are initially unique. They express a unique TCR sequence, But as they migrate or differentiate along the trajectory, they become clonally amplified. And at the end, when they are these exhausted T cells, it is very well possible that you end up with 10 to hundreds of these T cells with exactly the same TCR sequence, recognizing exactly the same tumor antigen. Now, while doing these single-cell studies for many years, I think that the most striking example of how single-cell can really help in understanding how tumors respond to therapy are actually in the field of checkpoint immunotherapy. So here you see how in normal untreated tumors, up to 8% of the T cells are clonally amplified. When we then treat or boost these tumors with one dose of checkpoint immunotherapy, in this case pembrolizumab, you see that after 1 week already, up to 32% of these T cells become clonal. And this basically indicates that there is actually a huge local proliferation of these T cells in these tumors. And this process is referred to as T cell expansion. And this is the process I would also like to discuss during my talk. So just a couple of key questions I would like to discuss regarding these expanding T cells. So the first question is, which are these T cells that are expanding and where in the tumor do they reside? So to answer these questions, we started a BioKey clinical study. This is a window of opportunity study in early breast cancer. And basically what happens is that we take a needle biopsy. The patient is treated with one dose, one boost of checkpoint immunotherapy, and then the patient undergoes normal surgery. And so both biopsies, the pre- and the on-treatment biopsies, are subjected to single-cell RNA-seq and TCR-seq. So here you see this result. So the heterogeneity within the T cells, you see PD-1 expression in two subsets, the CD4 positive T helper 1 and the CD8 T exhausted subset. And when we now look at clonal T cell expansion, so here we color each TCR sequence with a unique color, you can very clearly see that there is unique and quite obvious expansion in the two clusters that express PD-1, namely the CD4 T helper 1 and the CD8 T exhausted subclusters. And this is good because we see T cell expansion both in the CD4 and in the CD8 T cell compartment, which is supposed to sustain or to lead to a good immune response. Now, this is not true for all the tumors. We also did a window of opportunity trial in head and neck cancer. So here we treated the patients with anti-PD-L1, the first 10 patients, the next 10 patients with a combination of anti-PD-L1 with anti-CTLA-4. Again, we submitted both biopsies to single-cell profiling. And what we found is that in the anti-PD-L1 arm alone, there is only CD8 T cell expansion, which you can see here. When we then add on anti-CTLA-4, we basically see that also these CD4 T cells start to expand. So this basically means that depending on the tumor type, but also depending on the treatment, you get actually expansion of different T cells, being either CD4, CD8, or the combination of both. The next question is, where are these expanding T cells located in the tumor? I explained to you that we have up to 72 immune cell subsets within the tumor microenvironment. And what we can do is we can basically correlate the abundance of each immune set with the abundance of the other immune sets in each tumor. And what we then see is that there is actually two modules which each consist of immune sets that are very strongly and positively correlated with each other. So in the first subset, or in the first module, we see that there is CD4 and CD8 exhausted T cells. There is regulatory DCs, CD4 regulatory T cells, inflammatory macrophages, suggesting that this module is actually involved in type 1 immunity and is actually the microenvironment where these T cells are reacting against the tumor cells. And this is indeed the case because when we correlate this module with T cell expansion, we see that there is a positive correlation. There's also a second module, and this module consists of B cells, plasma cells, follicular T cells, and this is what we think represents the tertiary lymph node structures in these tumors, and indeed there is also a positive correlation of these structures with T cell expansion. Just as a confirmation, so here for the first module, the type 1 immunity module, we basically stain expanding T cells using the Ki-67 marker. And what we see indeed is that most of these proliferating, expanding T cells reside in such a type 1 immune environment and are either CD4 positive T helper 1 cells or CD8 positive T exhausted cells. Regarding the second module, so the tertiary lymph structure model, we confirm that these consist of B cells, plasma cells, and immune regulatory T cells, DCs. But there is not so much T cell expansion ongoing in these lymph node structures. What we think is that something else is happening, and what we actually found is that in these tertiary lymph node structures, there is actually a lot of maturation of the B cells, and when we perform single-cell BCR sequencing, we can actually identify also that there is expansion of B cells, mainly in the plasma cell compartment in these tumors. What is also very interesting, that is that when you correlate T cell expansion with B cell expansion, that you see that they are both strongly correlated with each other, suggesting that it's not only the T cell expansion, but also B cell expansion that could be contributing to the response. Now up to the next question, and that is whether this T cell expansion that we observe after one boost of checkpoint immunotherapy, whether that T cell expansion also leads to clinical response several weeks or months later. And for this, we actually got access to samples from the Neo-Immunoboost trial in collaboration with Peter Fasching from Germany. And so this trial is actually in triple-negative breast cancer, and patients actually receive the keynote regimen consisting of pembrolizumab combined with paclitaxel and later on epiribicin with cyclophosphamide. And what is special about the Neo-Immunoboost trial is that these patients receive a boost with anti-PD-1 prior to the keynote regimen. And also there is actually 4 biopsies available from each patient taken at different time points. And this now allows us to investigate whether T cell expansion, which we determine with these 2 first biopsies, before and after the boost, whether this T cell expansion leads to PCR or no PCR later on. So what we did is TCR sequencing. We looked at clonality, comparing the after boost to the baseline samples. We identified a subset of patients where there is very clear increase in clonality after the boost, and these patients, we refer to them as expanders. The other ones are non-expanders. And what we see is that indeed 8 out of 9 patients that have expansion, that they also develop a PCR. There is 1 patient that developed a no PCR, and we currently still need to investigate whether this patient maybe had an intermediate response. The other 7 patients in the PCR group, we think that they responded to chemotherapy. So in other words, it looks like T cell expansion very early during checkpoint immunotherapy also translates into PCR later on. The same can actually be appreciated here. So here you see the clonality changes over the 4 different time points in the non-expanders and in the expanders. You have this sharp increase in clonality after the boost. But what is interesting then is that this increase in clonality decreases as therapy continues. And what we think that happens is that actually this T cell expansion is a very early phenomenon taking place when you boost the tumor with anti-PD-1, but that then later on during treatment, this expansion of the T cells disappear. We think that this comes because bystander T cells in the tumors are also proliferating, but this of course doesn't last during the full treatment. What is interesting is that we also did bulk RNA sequencing on these tumor biopsies, and here we did a differential gene expression analysis in the expanders that develop PCR, comparing after boost with baseline samples, and this very clearly identifies more than 50 T cells involved in T cell activation. So clearly this expansion is also activating the T cells very strongly in these expanders. In the no PCR group, this effect is completely absent. So these T cells show no sign of T cell activation. In the non-expander group that is also developing a PCR, we see more or less an intermediate effect. So there is still some T cell activation, but clearly it's much less stronger than in the expanders. So what this suggests is that maybe we can look at T cell expansion after the boost to separate no PCR from PCR patients, But then what it also suggests is that this TCR expansion can actually be used to select a subgroup of patients that is actually responding more clearly to the anti-PD-1, whereas the other patients might be more responding to the chemotherapy. Okay, so for now I've been discussing how T-cell expansion can be used by profiling 2 tumor biopsies, one before treatment and one after treatment. But this is, of course, not feasible in a clinical setting because taking on-treatment biopsies is, of course, very invasive. So the question is whether we can measure T-cell expansion now non-invasively. And so for this, we found, again, access to the BioKey trial, window of opportunity early breast cancer, 1 boost of anti-PD-1, but we also collected PBMC samples before and on treatment. And so first we investigated whether we could detect T-cell expansion in the PBMC samples that we collected from these patients. But the answer here is no. So when you see very clear differences in single-cell TCR data, but also in single-cell RNA-seq data in the tumor, in the PBMCs you cannot detect this ongoing T-cell expansion. Another thing we then tried was to basically look at TCR sharing between the PBMCs and the tumor. And the reason for this is, of course, because there is a lot of T-cell trafficking from the tumor to the tumor-draining lymph nodes and back. And what we observed is that, indeed, there is actually more sharing of TCRs between the tumor and the PBMCs in the expanders compared to the non-expanders. And when we further stratify this according to time points, so before or after the boost, we see that this increase in sharing in the expanders is actually mainly coming from the on-treatment sample. What is also interesting, that is that if we look at these shared TCRs in the expanders, that we see that 25 to 30% of them is actually also TCRs that are expanding in the tumor. So in other words, by focusing on the shared TCRs, we are indeed looking at expansion that is ongoing in the tumors. We then also try to replicate this in the neo-immunoboost trial, and indeed, what we found is that there is an increase in TCR sharing in the patients that have pathological complete response, and this sharing then decreases later on during treatment, consistent with the effect of the T-cell expansion that we observed earlier. And finally, a question is, of course, we see this T-cell expansion. We think that these T-cells are recognizing tumor antigens on the triple-negative breast tumors, but we actually don't know which are the antigens that they recognize. So the question is whether we can, for these expanding TCRs, can identify the tumor antigens. And so for this, we have recently developed an in vitro platform where we try to identify these tumor antigens. So the first thing we do is actually in tumor biopsies from patients identify by TCR-seq the TCR sequences that expand. Once we have identified those, we clone these TCR sequences in a Jurkat T-cell line that is engineered in such a way that when the TCR recognizes the antigen, there is actually GFP expression also in the T-cell line. Then we take tumor cDNA from the tumor and we transduce this in an antigen-presenting cell line. Now, this antigen-presenting cell line is again engineered in such a way that it lacks the endogenous HLA, but actually expresses the patient-matched HLA from the patient we are investigated. We then also transduce tumor cDNA in this antigen-presenting cell line, and overall we know that when we do this in 10 million of antigen-presenting cell lines, we basically represent the full transcriptome that is expressed in these tumors in these antigen-presenting cells. And then we combine, and so what I forgot to mention is that whenever there is a transcript integrated in these antigen-presenting cell lines, that we have a red fluorescent protein. So then we bring the two together, the T cells with the antigen-presenting cell lines, and then we screen for a red and green signal, basically indicating that the TCR is recognizing the antigen. And this actually works quite well. So we now have a whole system set up where we can automatically detect the red and the green cells. We can amplify them in vitro, and then using fungal sequencing, we can basically identify the sequence that gives rise to the antigenic peptide that is recognized by the TCR. Just to give you an example, so here is a TCR28 that we identified in a BioKey patient. So this TCR was expanding. It was an exhausted or an effector T cell, indicating that it was cytotoxic and very active. And what we found is that this TCR actually recognized a cancer testis antigen FTH-L17. And here we see the corresponding single-cell data of this patient. And indeed, this gene is specifically expressed in the cancer cells from this patient. We also identified the HLA that is presenting this antigen. So this is HLA A0101. And what we also identify is that then this peptide can actually be recognized by the TCR sequence in vitro. Now, what is interesting is that this gene, FTLH17, is actually almost not expressed in normal tissues. We mine public databases for this. But when we look at the TCGA data, you see that there is quite a lot of tumors that are expressing this potential antigen. Here we mined the TCGA data a bit better, and what you basically see, looking at different thresholds, is that more than 2% of tumors in 10 different cancer types are expressing this antigen, and specifically in breast cancer, 2.7% of the tumors are expressing this cancer antigen. So this seems to be a pan-cancer tumor antigen that is actually recognized by the TCR that we identified in the single-cell data. Finally, what is also interesting, I think, is that this gene is very lowly expressed in normal tissue, actually lower than other pan-cancer tumor antigens, such as MAGE-H3. So finally, as a conclusion, I would like to, or I hope that I have convinced you that single-cell TCR sequencing can identify T-cell expansion as an early response marker to checkpoint immunotherapy. I've shown data that different T-cell populations can expand, and that this depends on the combination of the treatment and the cancer type. Remember the breast versus the head and neck data. A type 1 immunity and a TLS microenvironment seems to be sustaining this T-cell expansion. And also what is important is that T-cell expansion is a transient phenomenon that can translate into PCR and neoadjuvant triple-negative breast cancer. TCR sharing can be used to monitor T-cell expansion non-invasively. And finally, what I think is very interesting, that is that these expanding TCRs also offer possibilities to identify pan-cancer tumor antigens. And this, of course, has potential for vaccination and TCR-based therapies. And with this, I would like to thank you for your attention. I especially would like to thank the many clinicians that have been involved in collecting all these tumor biopsies from all these different clinical trials, because these are really key for our studies and for driving the findings that we make. Thank you.
Thanks a lot for this exciting presentation. So now we will move to the third presentation of our session, and this will be given by Dr. Christos Sotirou. Dr. Christos Sotirou is currently a research director at the FNRS, chef de clinique at the Institut Jules Bordet, and also recently was appointed as the director of the Bordet Cancer Research Laboratories. He's also directing the Laboratory for Translational Breast Cancer Research in that institute, and with his team, he focuses on improving the molecular understanding of breast cancer biology, disease dissemination, and progression by using different state-of-the-art omics technologies. In his talk now, he will talk about how spatial transcriptomics technology can be used to characterize the heterogeneity of triple-negative breast cancer. Christos, thank you.
Thank you, Christine. All right, so let's now change the topic and discuss how spatial transcriptomics can help us to better understand, particularly focusing on TNBCs. Those are my disclosures. Here's the outline. So first I give a brief background on TNBCs based on bulk RNA sequencing that we all know. And then I will present the data on using spatial transcriptomics, how we can dissect this disease and hopefully better understand and come up with a better treatment. And I will finish with some conclusions. So I guess many of you knows that when we're talking about triple-negative breast cancer, it's not just lack of ER, PR, and HER2. This is based on gene expression. There are at least 5 to 6 different subtypes based on their gene expression using the bulk RNA, not single cell or spatial. These authors at time more than a decade, they defined the basal-like tumors, the immunomodulatory, the mesenchymal, mesenchymal stem-like, and LAR. LAR is the triple-negative breast tumors that express androgen receptor. They look more like luminal tumors compared to the other triple-negative breast cancer. So after that, we also look at the mutation landscape that may characterize these different TNBC classes. And also here shown in the red arrow, the LAR, the luminal androgen receptor subgroup, shows the highest number of mutations in for instance, PI3, PIK3CA, AKT, and others. Actually, this is the subtype that shows higher somatic mutations, as we see in luminal tumors in general. We also found that, for instance, the luminal androgen receptor are the ones in the high, 75% of those patients may include mutations, the PI3K, AKT pathway, that today we neglect in terms of therapeutic options for those patients. And finally, looking for different single pathways, as an example here, we showed that, for instance, the mesenchymal stem-like tumors, they're the ones showing higher genesis pathways as compared to the basal and LARs. That again, we neglect those information and we don't do anything about that. We also knew, and this is also historical data, that within the triple-negative breast cancer, the ones that present high number of TILs, they have a better outcome. And in that paper, we had demonstrated that indeed triple-negative breast cancer have showed higher number of TILs as compared to the to luminal tumors, for instance. But also, it doesn't matter. It's important to have TILs, but not only counting number of TILs, but where and how they are localized. This is a very interesting study that has been done by a Canadian group that they microdissect the CD8 tumor cells, and they derive the signatures that can actually capture 4 different patterns of how the TILs are within the tumor. What they call fully inflamed are those tumors that have TILs all over the place in contact with tumor cells. The stroma restricted pattern that TILs are located in the stroma, the margin restricted TILs are located outside of the tumor bed, and then the immune desert that we don't see any TILs. So we took those information, and here again, looking at the bulk RNA sequencing data, we demonstrated, for instance, that immunomodulatory tumors, basal triple-negative tumors, they have this full inflamed phenotype, TILs everywhere in the tumor. The basal are the ones having TILs within the stroma, whereas the mesenchymal stem-like, and LAR are the ones having the stroma restricted or and some of those immune desert. And you see here on the right in the Kaplan-Meier curves that despite all of those tumors have a high number of TILs, actually the ones who showed a higher, better survival are the ones having this full inflamed phenotype, the TILs that are in contact with tumor cells. So now, what is spatial transcriptomics? This is very, very fancy technology that allows now to interrogate the gene expression in the context of the spatial organization in the tumor. So we can actually interrogate what are the RNAs that are expressed, for instance, in tumor cells as compared to stromal cells in different locations within the tumor tissue. This is a very easy technology. It's very expensive. So you take your tissue, you put on a specific slide that it has 1,000 of probes. The RNAs are going to link and attach to those probes, and then we will interrogate and we'll amplify those RNAs, and we'll have the gene expression in the context of their special organization, location of the tumor cells and stromal cells. So having this technology, we then asked the question, just here. The question now, acknowledging this in TNBC classification based on the bulk RNA, which is actually a mixture of tumor cells and stromal cells, can you use this technology to better characterize this TNBC classification in those different types? So the question here is that do molecular TNBC subtypes display distinct morphological patterns? What are the cellular biological processes shaping the architecture that characterizes each TNBC subtype? does the TLS composition which is the organized, immune organized response within the tumors, matters in the prognosis and response to immunotherapy? And can you actually improve this classification? Having this spatial organization of the tumor can improve their TNBC classification beyond the TNBC subclasses. So here is the workflow that we took now a few years ago. Everything is published in, again, 10 days before, actually, in Nature Communication. You can go and read in more details. So here we've had a study about 100 TNBC samples. We had some HNA slides to do the spatial transcriptomics. We also used the double immunostaining for CD3 and CD20 to pick those TLS structures, and also we have done some bulk RNA sequencing to compare our spatial transcriptomics with the bulk RNA sequencing. First of all, I would like to just show you how, in terms of morphology, our TNBC tumors are. You see here that it took us a year actually to annotate all those different morphological annotations, right? Meaning tumor cells, stromal cells, inside to vessels, tertiary structures, etc. So it took us a year, and you can see here from these images that we see that every single tumor is different from to the others. We found also that, according to the classification, that you see the difference in terms of these morphological annotations. For instance, the immunomodulatory tumors showing higher tumor content. They show higher number of TILs. Of course, that's why they were classified as being immunomodulatory. And in contrast, the mesenchymal stem-like tumors in large are the ones that are having the lowest in tumor content, the higher stroma content, and there are higher number of fat tissue, which actually just simply by looking on those histological annotations, you can actually discriminate those different TNBC subtypes. I guess that AI algorithm in a year from now, they will be able to do that in a second. We also found that, again, looking on the morphology, just looking at the morphology, that there are some differences. For instance, the basal-like tumors and the immunomodulatory, they grow in big patches, as you can see here, as compared to the LAR and the mesenchymal stem-like tumors that are grown with thin, small patches and they're more scattered in the tissue. Again, different patterns in terms of how those tumors are growing that can just depict by just looking on the histological annotations. Now let's move to the gene expression, having this RNA data from the different histologies and morphologies. We asked the question, what is the contribution, for instance, according to this, the contribution of stroma, for instance, in order to say that that tumor is immunomodulatory, that tumor is mesenchymal, and so on. So here's the alluvial plot. It's a little bit complicated, but just go through the slide. So this is in the middle is what the classes of our TNBC tumors according to the bulk RNA. So everything mixed, all right? Tumor cells, stroma cells. So we have the LAR, we have the mesenchymal stem-like tumors, the mesenchymal, the basal, and the immunomodulatory. Now, if we're looking only on tumor cells, because you have this possibility, you can actually interrogate from our data, give me all the RNA data that comes from only tumor cells, no stroma cells. So you see here that we have actually only 3 classes. So instead of having 5 classes, because the mixture of stroma and tumor cells, you have only 3 classes if you interrogate only tumor cells, namely the LAR, the mesenchymal, and the basal-like tumors. Here there are some contamination because we are not in the single cell level. And also it means that when we say immunomodulatory, our basal-like tumors having a lot of TILs, a lot of immune cells within the stroma, and having this full inflamed pattern. So when we say immunomodulatory, it means that you have a lot of TILs, you have basal tumors, a lot of TILs everywhere in the tumor. When you have a basal-like tumors based on your bulk RNA, are basal tumors cells that have TILs now, not in the having the full inflamed phenotype, but more in the stroma restricted phenotype. You see that the mesenchymal stem-like tumors doesn't exist. There are mesenchymal tumors with a distinct stroma, with different stroma that make them to be mesenchymal stem-like according to the RNA, to the bulk RNA sequencing. And LAR are very, very different. So, and you see that on the right, that if you now interrogate what are the differences in the stroma, there are some differences. For instance, you see obviously that the immunomodulatory shows a very high expression of immune cells, of course, and any signatures that shows immune activation. In the opposite, in the large tumors, they are actually depleted on any of those immune signals. You see that in mesenchymal tumors, you have some neutrophils in the stroma that don't have in other subtypes. In contrast, in the mesenchymal stem like tumors, you see some CAFs that are more present. You have some more angiogenesis. And you see actually the mesenchymal stem like tumors that are between in terms of the immune expression. Between the immunomodulatory having the highest expression and the large mesenchymal having the lowest expression of immune genes. And another example here is now if you're taking the mesenchymal tumors based on the BAC RNA, you see that if you're having different stroma components that you cannot actually depict from the RNA BAC sequencing, you see that those mesenchymal tumors having this mesenchymal cell-like stroma, they have a very, very good outcome as compared to the mesenchymal tumors having the mesenchymal stroma and having a poor outcome. And this was basically mainly due to the more immune signal within this mesenchymal stem-like stroma as compared to the mesenchymal stroma. Now, the next step, we were very interested in the intercellular lymphoid structure because I'm sitting next to another lab in my institute directed by Karen Willard-Gallo, that she was the first actually years ago to demonstrate the presence of those intercellular lymphoid structure within breast tumors and how important it is in terms of prognostication and probably in terms of immune response. So we took this advantage, having a big number of tumors, having this possibility to dissect in silico those TLS within the tumor. And this is what we've done here. So we have the H&E staining, we had the histological annotation from our pathologists for this TLS. We had also the immunochemical staining for those TLS. And then our brilliant bioinformatician, actually, who was the senior bioinformatician in the study, came up with some regression model to be able to capture the RNAs within each of the spots that we have on the slide that could predict the presence of TLS. And actually, he did that for any other morphology within our slides. So by doing that, so we were able to derive any expression that is present only in TLS, but not in TILs, not in any other immune compartment that is not organized as a TLS, as well as within TLS, but not in the other non-immune microenvironment. And if you see here, by doing the Excel deconvolution, many of those cells make sense to be part of the TLS structures, some for instance, B cells and others. And we were able also to derive 30 gene signatures by doing the same exercise, everything that within those structures, not outside of the structures, for instance, in TILs that are in the stroma or outside the stroma, and everything that is not immune and stroma, and we come up with these 30 gene signatures that, if you're looking for the gene ontology, many of those genes are associated with angiogenesis, proliferation, B cells, and germinal centers, which again makes sense to capture this organized immune response. Of course, the next step was to see whether what we found makes sense, and this is showing here how nicely you can map those 30 gene signature on those TLS structures that you can see either by immunohistochemistry or by the annotation or by our regression model. Next step is to see whether, before that is as expected, the expression of that TLS signature was higher in immunomodulatory tumors, the ones that have this full inflamed phenotype, and in contrast, the mesenchymal and large, the ones that are showing the lowest expression level of that signature. Also correlates very well with the time classification. The tumors having this full inflamed, they're the ones having more expression of that TLS signature. And also, if you're looking now for the distance of those TLS structures within the tumor or outside the tumor, we see that the expression of that signature was highest in those TLS that were within the tumor bed. Now, in terms of response, of course, this is something that we looked, looking now at the public available data set. You see that the highest expression of the TLS signature shows a better outcome as compared to the tumors, showing the lowest expression. We also went to the I-SPY data set to see whether it can predict immune response, and you see that the highest expression level was associated with higher PCR rate in the I-SPY. And also we were able, since the TLS are not breast, it's just the host environment, we went to the data sets with, that were treated with immunotherapy only in other tumor types than breast, and we demonstrate that indeed that the signature was the one of the best signatures predicting immune response, and this was true after correcting for many other immune signatures, including other TLS signatures that have been valued by other groups. All right, now, the next step that we wanted to see is that having this fancy technology and all this data, can you do better than TNBC classification? Can you characterize our triple-negative breast tumors better than that? So here we have done this analysis based on 3 different steps. So this shows you here the number of clusters. And so one cluster means that you have the expression pattern that resemble. It can be within the tumor, can be within the stroma, or can be in combination stroma and tumor. So we found about 5 to 7 different clusters per tumor. In our data set, we found about more than 400 of those clusters, but many of those clusters were shared among patients. So you come up with 14 megaclusters. So there are, again, gene expression patterns that capture this architecture of our TNBC tumors, shown here. On the left, you can see here how those megaclusters capture the different morphologies. For instance, the top 5 clusters are associated with more tumor content, less stroma, whereas those megaclusters are showing more stroma genes than tumors. You see, again, the LAR subtype shown here in red is very unique. It's very different from the other subtypes, and some of those were associated with survival. Now, many of those megaclusters also are shared within patients. And this is what we come up with, what we call now archetypes. We come up with 9 archetypes. Each patient may have up to 7 of those megaclusters, and the archetypes shown here. So here you have the 9 archetypes. On the bottom here, you can see the different TNBC classification as well as the timing classification. You see here that, and here the prognosis. You see now, interesting, for instance, that there are 2 archetypes that shows tumors having the full inflamed phenotype with a lot of TILs. But you can see, despite the fact that have all high TILs, there is only archetype 4 showing a better outcome as compared to archetype 3, that despite having high TILs, they still doesn't have just a good outcome. Again, you see here, this archetype 5 was mainly seen in LARS. Again, LARS, the luminal adjuvant receptor, it's very, very different from what we think basal and mesenchymal tumors. And shows here some characteristics of those archetypes. The 4 first archetypes shows a lot of immune response. Archetype 4, which having the better outcome, shows also the presence of TLS, organized immune response, having highest expression of our TLS signature. Archetype 5 shows a lot of metabolic genes that are characterized in LARS. Archetype 8 here, there is depleting in immune signal, but we found here the high expression of Nectin-4. We understand that, at least this is the hallmark of that archetype. And here are summarized also some surface markers that are important now for the ADCs that we treat for our patients, for instance, TROP2, RBB2, and Nectin-4. We see that not all archetypes are expressed at the same level and with those receptors. For instance, there is the archetype 2 that expresses in TROP2 and Nectin-4, archetype 5, which is mainly LAR, expresses high level of RBB2, and we know that LAR, this androgen receptor, was shown previously high expression of RBB2 and etc. This is a final slide that's summarizing the protection precision medicine for TNBCs. Of course, it's the beginning. Shows you here how these archetypes are more granular than the TNBC classification based on the bulk RNA sequencing. And there are some vulnerabilities that we may actually think to follow in clinical studies, such, for instance, the archetype 4, having this organized immune response. Maybe they are the ones who maybe do not need to give any treatment. There's the ones that are having these high TILs, having this full phenotype has organized TIL response. Maybe those are the TNBCs, at least for lower stage TNBCs that maybe actually de-escalate. The others, archetype 2s, are ones that probably need to give chemoimmunotherapy. LAR is very, very different from the others. We have to think about combining anti-androgens, combining metabolic drugs, combining PIK3CA inhibitors and CDK4/6 inhibitors. There are ongoing studies for those LARs, and I didn't say that, but the majority, not majority, but there's enrichment of lobular tumors, triple-negative lobular tumors having this LAR phenotype, and etc. So to summarize, I hope that I convince you that TNBC is not just only lack ER, PR, HER2, it's more complex than that. And they have distinct tumor organization and cell composition. There's a substantial interpatient heterogeneity within those tumors. We derive at least 9 archetypes that capture that spatial organization of TNBCs with different vulnerabilities, and we derive a signature that can be actually be used in order to identify patients, not only for breast cancer, for any other tumor types that may respond to immunotherapy. So with that, thank you very much. This is an acknowledgement. This is the people that were actually involved, some of them, to the study, our founders, and with that, thank you very much for your attention.
Thanks a lot for this beautiful presentation. And so we will now have our last presentation, which will be given by Dr. Emanuela Romano. So Dr. Romano is actually a physician scientist specializing in oncoimmunology, breast cancer, and tumor immunobiology. Since October 2015, she serves as the medical director of the Center for Cancer Immunotherapy at the Institut Curie in Paris, where she has already led the development of academic trials of immunotherapeutic modalities in breast cancer, where the trials have also a strong translational components. She authored several publications on the tumor microenvironment in breast cancer and in other cancers of the women. Here, she will discuss how single-cell RNA sequencing of tumor-infiltrating lymphocytes can be a powerful tool that provides with gene expression data at the level of individual cells and can be used to determine factors associated with treatment outcome. So, Dr. Romano, the floor is yours.
Thank you for the nice introduction, and thank you for being here until the last talk. I guess we will switch gear again to single-cell RNA sequencing, and I will try to explain how we leverage the potential of this new technology to define a potential biomarker of response to chemoimmunotherapy in metastatic triple-negative breast cancer. So this is my disclosure. None is related to the talk of today. So one of the major limitations in cancer immunotherapy, I think we all agree, is the absence of patient selection based on target expression. We typically in the clinic propose a clinical trial. We run a number of tests, scans, checks, blood checks, and tumor biopsies, but the day of treatment really we are not sure that we are hitting the target and we are really producing potentially a clinical benefit for the patient. And there are several reasons to that. Conventional tools that we use normally in the clinic don't fully address these key questions, and we don't really assess precisely target expression nor the cell subset engagement, for instance. And we rely, for instance, on immunohistochemistry that gives a semi-quantitative evaluation of a given target in a spatial scoring. But the point is that this is relying on fixed tissue with the caveat of using fixed tissues. Sequencing, typically bulk RNA sequencing, is basically a a smoothie of a much more rich sample. So we need massive deconvolution to really depict the tissue of origin. That's why it's interesting to look at single-cell RNA sequencing, because over the last decade, we actually witnessed several efforts to arrive to capture the mRNA of the single cell. And in 2015, we started to see coming up the first publication looking at a technology whereby single cell was captured in a lipid droplet, and be able to be sequenced for the mRNA content. And 10x Genomics was actually one of the companies, the most successful, I guess, that could implement this technology more broadly across institutions and countries. And so basically, the number of cells that one can study has grown from a handful to over 10,000 cells in 10 years. And also for us immunologists, it's very important because this technology allows not only the resolution of the single cell subset, but also the state. We are interested to know, of course, whether a T cell is a CD4 or a CD8, but we also want to know whether this is an effector cells, whether they can make gamma interferon or things like that. And this is possible with this technology. So very briefly, 10x RNA sequencing basically start from normally tissue dissection and a cell suspension, which is then captured with droplets. So we call this cell partitioning. And this essentially allows the capture of mRNAs in a microfluidic channel. Then a library is prepared, which contains basically BSC and UMI of the single transcript, and then essentially sequencing is done, and the analysis can be performed. I wanted to, since this is a workshop also, I wanted to bring up to your attention some caveats, limitation of this technology, because as any technology, there are some limits. We know that only the 3' end is sequenced normally if we want to use the short read. In this study that I'm showing you, we also looked at the 5' end for TCR sequencing, but it's a more cumbersome, let's say, sequencing. You can, as of today, sequence maximum 10K cells, but with the newer device, perhaps we can go up to millions. There are some limitations about the cell size, because when the cell is too large, it can actually clog the microfluidic channels. And also you may have droplets that contain, for example, 2 cells. It can be interesting now. There are several publications that show when you do cell-cell contact and cell-cell interaction, actually doublet can be a readout of cell communication, but normally we exclude doublets. And also some practical information for the experiment, because I think we were confronted in the lab on these kind of questions. We use fresh cells, and for this it's very important. The time between dissociation and the actual 10x experiment is less than half hour, less than 30 minutes, because otherwise you start collecting mitochondrial genes, stress genes that are not really interesting and were not essentially expressed in the original tissue. And the frozen cells can be a nice idea to overcome the limitation of the fresh cells, but unfortunately, at least in our end, but not only in our end, for immune subset doesn't work really well. In our opinion, it's mostly working on cancer epithelial cells. And obviously, you have to run a bit more cells in order to capture the 10,000. And also, another thing is that some optimization have been done, for example, on PBMCs that are naturally in suspension. They are immune cells, but the results do not really reproduce what is on cell dissociated from the tissue. So as you understood, we've used this technology to ask a question about a triple-negative breast cancer patient. undergoing chemoimmunotherapy. TNBC is diagnosed in about 15 to 20% of breast cancer patients, doesn't respond to hormone treatment or HER2 targeted therapies, and when it's refractory to treatment, is has quite dismal prognosis. Today in the clinic, we are able to offer chemotherapy plus pembrolizumab, an anti-PD-1, in first-line treatment for locally advanced or metastatic breast cancer patients that whose tumor express PD-L1 at a CPS score of at least 10, which is good because this trial, the Keynote 355, showed advantage in overall survival compared to chemo alone. But we all agree, I think, that we would like to increase upon this benefit and increase the area under this yellow curve. So there are several hypotheses that have been made and strategies that have been tested in the clinic to which other biomarker or other signaling pathway can be interesting to target in these cells. And so one of those that are in active investigation right now is the adenosine. The adenosine is a catabolite of the ATP that is catabolized in AMP to adenosine, a soluble mediator in the tumor microenvironment, via two ectoenzymes, the CD39 and the CD73. And the adenosine has several immune suppressive properties in terms of antigen presentation, innate immunity, the cytolytic capacity of NK has been demonstrated to be disrupted, but also the adaptive T-cell immunity. And there are several drugs that actually target this pathway at several levels, the CD39 or the adenosine receptor or the CD73. And the colleagues, actually no, and another point is that the CD73 expression at the gene expression level or at the protein level has been also associated with poor prognosis in TNBC. So for this reason, the colleagues at Jules Bordet designed a phase 2 randomized trials where essentially first-line triple-negative breast cancer patients were offered a trial whereby they would receive a chemo doublet, paclitaxel plus carboplatin, with either an association of oleclumab, an anti-CD73, and durvalumab, an anti-PD-L1, or the same doublet plus durvalumab. And the primary endpoint was the clinical benefit rate, so stable disease, complete response, and partial response at week 24, so by 6 months of treatment. Unfortunately, the study was negative for the primary endpoint because the clinical benefit rate was not statistically different in either of the arms. We did see some response, obviously, and so it was very important to us to understand which of the patients were responding to this treatment and which were not. And so, thanks to a collaborative effort between my institution, via Leuven, and also Institut Bordet, we were able to collect about 30 samples from this patient recruited at our institutions at baseline and on treatment at week 3 of patients that were either responding or not to treatment. And what we did, essentially, we sequenced by single-cell RNA sequencing, and we focused, at least in this study, on the immune compartment, particularly on the T-cell compartment. And here you can see that you have 12 clusters of T-cell, the CD4 compartment, and the CD8 compartment. So, and they span the various states of the CD8, for instance, from more progenitor than to more dysfunctional late effector cells. Here you can see, for example, the CCL13 clusters is mostly found in the late effector, whereas the GNLY or the CCL2 more in the early effector clusters. And then we asked a question about whether there was a difference in the fraction of these CD8 T cells that were able to discriminate between responder and non-responders. And what we saw is that there were higher fraction of these more exhausted cells, the TOX or PD-1 overexpressing in the responder. In particularly this CD8-6L13 cluster. These cells are late dysfunctional cells in the sense that they arrive to the late stage of the adaptive immune response, meaning that this patient experienced an anti-tumor response and were expressing, for instance, PDCD1, anti-PD1, LAG3, so all markers, TOX, all markers of exhaustion. Together with 6CL4, there is an early memory marker, and that is associated also with TCF1, as Raza mentioned before, in responders. And instead, the no-responder had mostly a cluster of CD8 GNLY, so mostly TEMRA effector memory markers. And what we saw also is that the responders had mostly signatures of late effector exhausted cells. Interestingly, in the literature, there was already some observation about these cells, the CD8 double positive for CCL13, in anecdotic observation. Here you can see tumor-reactive T cells expressing, secreting CCL13, or in this study where the patient having PD-1 positive, CCL13 positive were responding to anti-PD-L1. And also a larger publication in 2021 showed that this subset of cells was predictive of response to immune checkpoint in general, anti-PD-1, PD-L1, or ipilimumab, anti-CTLA4. And so the next question that we wanted to ask is whether at baseline responders were also different from non-responder from a TCR point of view, because we know that TCR dynamics, as also Diether Lambrechts showed you before, are very interesting in terms of clinical outcome. And what we saw is that actually at baseline, responder patient expressed a number of clonotypes that were more expanded than non-responders. In particular, you can see also they had higher hyper-expanded clones, meaning from 20 to 100 clones expanded compared to non-responder, for example. And some intriguing observation that we made is that essentially the responder patient during treatment contracted this clonotype response, exactly like it was suggested before. And this we know, for example, even after viral response, we have this expansion phase followed by a contraction phase. And you can see also here in responders at baseline, they have several clonotypes that were expanded, but during treatment, these cells actually decreased. And so we wonder why this was the case, and we went back to our UMAP. We traced back these cells, the CD8 6CL13, the 6CL4, and we saw that indeed in responders, this 6CL13 indeed contracted during treatment, but they were kind of replenished by early effector cell, the 6CL4, for example, or the cycling cells. And in fact, in the cycling cells, there were a lot of genes that resembled this 6CL13. Instead, in the non-responder, we saw basically no TCR dynamic. It was quite flat, let's say. So, in conclusion, what I showed you is that in responder, in pre-treatment samples, we see an expansion of this 6CL13 CD8 cluster with a beautiful TCR clonal expansion, which could be tested by FACS, for instance, in a small panel. And also this is true on treatment with an emergence, and this is a new concept I think that we're bringing, of these new emerging clonotypes, like new, fresher cells, early memory cells. And in non-responder, this is not the case. They basically have mostly bystander CD8 T cells that are reactive to viruses. The GNLY I couldn't show you, but from signature, they're not tumor reactive, they're more viral reactive. And they have very little of these 6CL13 CD8 T cells. So in conclusion, I would say that this continuous replenishment of this late effector CD8 6CL13 axis is actually a proxy measure of response to checkpoint inhibitor associated to immunotherapy in advanced triple-negative breast cancer. And with this, I would really like to acknowledge the work of the people that did work in the lab, in the Institute, the mentors, and the various colleagues that made this collaboration possible, for their generosity as well, and the platform and department at Curie that made also possible all of this, and the funders, and you for the attention. Thank you.
Thank you very much for this also beautiful presentation. So what we will do now is that we will have 15 to 20 minutes of a panel discussion. So those of you who have questions, you can go to the microphone. I also received many questions also in the chat, so I will alternate the questions. So I will start by a question that actually could be answered by any of the speakers. So obviously, everyone was looking for markers of response or resistance to patients treated with a combination of immunotherapy and chemotherapy. However, it is very difficult, I think, to make the part of what are exactly markers of the combination of these therapies, what are markers specifically for chemotherapy, or what are markers for immunotherapies. So if maybe some of the speakers would like to comment on that, and also to comment on maybe a strategy to make to know which markers are actually for both, for only one of those, or for the two?
Yeah, maybe I can answer that question. How do you distinguish between markers which are markers of response to immunotherapy versus chemotherapy, for example? The short answer is have a randomized trial where some patients receive immunotherapy and the others receive chemotherapy. And so that was really what we were looking for in NEO-TRIP. Although all patients received chemotherapy, we were looking for markers which distinguished the patients who received immunotherapy. In the absence of randomized trial, it is difficult, but you could presumably compare between observational cohorts which do receive immunotherapy and others which do not, and determine whether a marker is predictive of response. But I think the gold standard remains true randomization, although that's obviously a very high bar and non-trivial.
Okay, so you want to answer that question as well?
I wanted to add, because it's a very interesting question, and I also think about it. I believe that we have some data that are recurrent. For example, we across the board show that late effector cells, TOX1, for example, are enriched in responser. You also and I show that TCF1 are also enriched in responders, and so TLS. So clearly, there is already a signature that we can test, even a retrospective cohort, for example. I think we are not that far, and perhaps this can be also implemented in the clinic with FACS analysis, for instance.
Okay, thanks a lot. So there should be a collaborative effort to look back, and specifically in randomized trials. I fully agree. We'll start with one question on the microphone on the left, and then we'll take questions there. Yeah. Please introduce your name also when you ask the question. Thank you.
Andrew Chat, ICR and King's College London. Thank you. Fantastic session. I'm going to be greedy if I can and ask a question first of Emanuela and Diether, and then maybe one of Raza. So to you both, fantastic talks. If I've got it right, you've shown us that those who respond to the addition of immunotherapy to chemotherapy are clonally expanding and then restricting that expansion, or then there's a focus of that expansion. So to me, the fascinating thing is, what is that expansion against? And I think Diether showed something about a cancer testis antigen candidate. Can I ask, in the B-cell expansions, which I think you showed, B-cell receptor expansion, do you show clonality there, and what is that directed against? Because those humoral responses could be generated as antibodies and used as therapy.
Yes, very good question, and I agree, of course. So we see B-cell expansion, so that's clonal expansion of plasma cells, what we showed. So these cells are antibody-producing cells. We don't know what they recognize. Actually, we have been thinking of setting up a similar screen as we do for the TCRs, but now for B cells, for plasma cells, and to try and identify what the antibodies they produce recognize. Besides that, these antibodies can also be specifically involved, for instance, in ADCC. So there is different mechanisms of how these antibodies can be involved. It doesn't have to be through recognizing epitopes, so to say. But it's a very good question. I've also been intrigued by this, so to say.
Thank you. And if I can ask Raza perhaps a question on your ability to analyze platinum in the imaging cytoph experiments. Have you been able to look at differences in the triple-negative patients in the platinum clearance from the tumor in those with BRCA defects, whether they're mutation or methylation, from those that do not have those? One might hypothesize it would be different. The dynamics of platinum in the tumor may change different.
Yeah, that's a great question. We haven't yet, because we haven't got the BRCA data. But in principle, it should be possible, and also to look in the way that you're suggesting, to look where the platinum is accumulating. What's interesting is it accumulates in particular in macrophages, but you do also see it in the cytoplasm of some cancer cells, and eventually in the extracellular matrix. So it may also be that where it distributes is different based on the genomic architecture of the tumor, and BRCA in particular. So yeah, that's a great question, something we should look at.
Okay, thank you. We'll move to the mic in the middle.
Hi, Gaurav Gupta from University of North Carolina. My question is for Dr. Lambrechts. You had posed the question of whether we can identify clonal expansion in blood, but then you said you have to match it to the clones in the tumor. But once you know which ones are reactive, is there a signature specifically of those tumor-reactive T cells in blood?
Yeah, very good question again. We haven't. I mean, it's kind of challenging to find back this signature in the blood, because the percentage of these shared TCRs, shared T cells, and that also expand, it's very small. It's 0.0 whatever percentage. So it's really a challenge to identify it based on gene expression. What I maybe forgot to stress is that we need to indeed identify the TCRs that are shared between PBMCs and the tumor, but the pre-treatment tumor sample is sufficient there. So you don't need to go and biopsy again in these patients. So that, I think, is kind of interesting, because with one tumor biopsy, it's sufficient.
Thank you. On the microphone on the right, yes?
Hi, Jake Layla from Texas Biomedical Research Institute. I feel like I want to kind of expand on the first question a little bit for Emanuela and Diether. So, did you ever check any of the head and neck tumors for viral reads, like HPV-16, which is found in over half of head and neck tumors?
Yeah, so they were all HPV negative, all these patients.
Interesting.
So,
Yeah, I just really, again, I want to answer the same question. Why are you getting a specific T response? And Emanuela kind of mentioned, oh, this is T cells that are in response to viruses. And so, but it was a really great talk by all the speakers. Thank you so much.
Thank you. We'll take the question again on the left.
Hi, Isabel Wall.
Yeah.
I'm a...
Yeah, no, it's for you, it's for you.
Hi, I'm a fourth-year PhD student at King's College London, also looking at TNBC using spatial transcriptomics. My question is for Christos. With your spatial archetypes, have you been able to identify the specific features of each of those archetypes that are driving those associations, rather than just the presence of given cell subtypes or pathways, but what, for example, interactions are specifically driving it?
So, I mean, there are a lot of details in the manuscript that I couldn't show here. So, yes, you look for several signatures of several cell types and states and etc. So your question specifically is whether
you've been able to identify what the specific driving features are. So if a given archetype is characteristic of a certain cell type, have you been able to identify what the signaling features are that are driving the association?
In terms of mutations, for instance, and so on. I mean, we haven't done that yet, but this is the beginning.
Okay, thank you. The microphone there, yes.
My name is Marleen Kok from the NKI in Amsterdam. Amazing session, amazing speakers, amazing data. My question is for Christos. I want to share a struggle with you, and you probably have the answer for me. So with the spatial transcriptomics platforms, you see that the amount of genes you can use is increasing. So and now you have a choice, if you have an interesting trial on, do you think you need, if you look at specific research questions for tumor microenvironment, that you need to go up to 3,000, 6,000 genes, also increasing the costs. And also, I've been told that if you increase the number of genes, you kind of saturate your slides, that certain transcripts, cannot be analyzed anymore. So what is your ideal number of genes for TME?
Yeah, I mean, you can ask the question to the panel. So the data I'm showing you here is the first generation of a spatial, which is not that detailed. I mean, we know now the Visium, we have more higher resolutions, and you have the single cell resolutions at the protein level, the expression level. They are now plotted from that. So I would say that we don't need to have all those 10,000. I mean, with a single cell level, with a couple thousand of genes probably is enough to detect and pick the most important information in terms of cell types and etc.
Okay, so I'll take a question from the chat, which I expanded a little bit. So obviously, all the speakers, they used quite some fancy and expensive technologies, we must say, which are not necessarily applicable to samples as it comes. So what would be, you can give all of you a short answer, what would be something that you would like to look at and which you think would be feasible in the context of the question that you want to answer? So I'll go... So Emanuela, maybe you can start, and then...
Yeah, it's a good question, and I tried to allude before in my talk. I understand, of course, we cannot run single-cell RNA sequencing on all, every biopsy that we have, but we can definitely run some FACS analysis, for example, of a fresh tissue biopsy with the collaboration of surgeons and pathologists to see, for example, if our subset is there, and so there are good chances that the patient may respond to this treatment. Okay. I think this is easily translatable in clinic.
So facts, that's the first answer.
Yeah, for me, my dream project would be to combine single-cell RNA-seq with spatial methods, but with spatial methods that really allow me to integrate the single cell with the spatial data at single-cell level, so that we can analyze the spatial data as if they are single-cell data.
Okay, but with a limited number of markers, or not
no,
to be
unbiased,
applicable?
no.
Unbiased,
So,
okay. Okay, good. Christos?
Yeah, so when you have this medical hat, I mean, I think understanding the disease is very, very important. And for instance, having demonstrated that, you have these TLS structures, and we don't need to have any spatial or any single cell, we just, look on histology. But now we know that this is very, very important, and it's number one. Number two, having this full inflamed phenotype. We don't need to have a mass spec. I mean, you see, on your eye, T cells, lymphocytes in conjugate tumors. That's probably this is the pattern that we found in your study, that those are probably the ones who respond to immunotherapy. I mean, this knowledge is important. And then of course, acknowledging that there are different CAFs, there are different neutrophils, macrophages. Then you can go back to the classical pathology, AEI, whatever, and based on this knowledge to go to the clinic. But I'm sure. I mean, there is no way to go with single cell spatial in the clinic one day. I mean,
yes. So also to use some already acknowledged markers like the identification of TLS or other markers which can be easily identified. Yeah. Raza, you want to give your...
What's the question?
So knowing that all the technologies that you all described and applied on all these data sets, based on the knowledge you generated, what do you think would be applicable actually in the clinic or could be expanded, to identify responders or patients who would be resistant to this treatment?
Yeah, that's great. I'll try to be brief. I think one of the nice advantages of the multiplex epitope-based imaging is that it uses antibodies and spatial proteomics. And clinical pathology essentially uses spatial proteomics. It's just not so omic-y. It's you use conventional immunohistochemistry. So at least in principle, it should not be the distance between that technology and classical clinical pathology is not as far as it is for some other technologies. Having said that, there are still some limitations with respect to specific reagents, particularly if you're going to try and conceive of them as companion diagnostics, where the regulatory threshold is very high. And so the other approach, which I think we should not discount, is the idea of identifying surrogate biomarkers using AI and digital pathology. I think that remains an enormous potential and might be a shorter route to getting some of these discoveries into a clinical setting.
Okay, thanks a lot. Very complimentary answers. I'll take the question further there from the third mic.
Yes, hi. Xavier Tekpli, also University Hospital. I have a question for Dr. Ali and Dr. Lambrechts, if that's okay. But first, I'd like to thank everyone for the amazing presentation, the great science, and amazing session. So I'll start with Raza, if it's okay. And I would like to extend a little bit on the resistant part and the CD15 positive cells. I went to the paper while talking, and I also saw another potential resistant marker or cell types, GATA3 positive. So I wonder if this CD15 and maybe GATA3, if you expand on them after, if they also have like the negative mark of sensitivity, so low MHC and low KI, for example, low proliferation. And next, if they are also having a specific immune microenvironment, are they having more immune cells or stromal cells expected by chance around them?
Yeah, that's a great question. Hi, Javier. So CD15 were the only ones which were really showed a significant difference. There were some others, as you suggest, which showed a modest difference, but it was really the CD15 positive cells where we were confident that this was a marker of resistance, and that was robust however we looked at it. Yeah, we looked at the time. I regret we didn't look at it in much more detail and spend a lot more time on trying to characterize their local TME. The initial analyses did not really throw up anything that was compelling, which was interesting. I mean, it really looked as though there was this one marker which was indicative of their resistance. And that was also in the other analyses. I mean, when we looked at the TME independently, we didn't see other markers of resistance where the two things would have been correlated. And so, yeah, it still remains somewhat difficult. It doesn't look as though they're orchestrating a local immunosuppressive TME. So my feeling is that this is a metabolic transition into an immunosuppressive subtype in a way that we don't fully understand yet, or at least we couldn't discern in our data.
But are you able to tell if they are actually MHC negative or?
Yeah, we can, absolutely, yeah. So we could just measure their expression level of MHC 1 and 2 relative to other cell types. Off the top of my head, I can't remember how it compared. I think we did look at that, so I'm guessing it wasn't terribly different.
Thanks. One more to Dr. Lambrechts, if it's okay?
Yes, a tiny question, because
yes,
after that we'll have only time for one question.
I'm wondering about the antigen that you're looking for in the assay. Do you think that proteogenomics can help identifying these peptides that are also, so it will be outside of what you're doing? And do you think that they need mutation also, these peptides? So now you talked about one, but do you think they need mutation to be actually driving expansion?
No, I think one of the advantages of the approach we take is that we can also identify antigens from the dark antigenome, like they refer to that. So we've identified some alternative splicing events, alternative promoter events that also are recognized by TCRs. But I gave this example in particular because it's actually a pan-cancer antigen, so there is multiple tumors that have this. So that's why I thought this was interesting.
And what about proteogenomics? Do you think it could add to your pipelines?
Oh, we have not really considered it, so yeah, I have to remain in the answer, sorry.
Okay, so we'll take one last question there on the mic, and then we will need to close the session.
Excellent presentation. You nicely described about the triple-negative breast cancer and the heterogeneity and how robust TCR expansion can be after immunotherapy. My question is more clinical. ER-positive disease, at least luminal B tumors, are just as heterogeneous as triple-negative in a different way. Do you see TCR expansion after immunotherapy in luminal B or luminal A tumors, or at least a transient increase in TCR expansion, if not lasting long expansion?
Yeah, so in this trial, the BioKey trial, there were also ER-positive patients, and we did see some T-cell expansion in some of these patients. So yes, there is T-cell expansion. We didn't really continue investigating other cohorts, but yes, we saw some.
Thank you.
Okay, so now I will thank the speakers again for the great presentations. I will thank you for your attention and the great questions, and enjoy the rest of the symposium. Thank you.